so today the plan is we're finishing up psychological research, and we're going to start what should be our unit this week, which is on biopsychology. So starting this week, it's going to be kind of those more individual topics in psychology going forward after we finish up here. Okay, and hopefully you're finding those seminars very helpful in practicing the material that you've learned. All right, so can someone remind me of our 3 big types of studies that we do in psychology? It was number 1 in your worksheet, if you remember the different types. Yeah, what do you got?
Experimental, correlational, and descriptive.
Yeah, great. So we have correlational, descriptive, and experimental. We said experimental is the gold standard because it's the only one that can tell us about cause and effect. Okay, so we did spend a lot of time on experiments. I do want to talk a little bit about threats to experiments because they aren't perfect, even though they are our gold standard. So I'll just name a couple just so you're aware of some of the limitations of this research design. All right, so number 1, they may lack a precise operational definitions. Okay. If you got to number 4 on that worksheet, that's what it was about, the operational definitions, right? So we need to make sure that the researchers are defining the construct, especially if it's abstract or conceptual, so everybody's on the same page. When the participant is filling out the survey or whatever they're doing, they're thinking of the same thing the researcher is thinking of. So very precise definition that's given by the researchers. Ideally, the researcher is not biased. They're going to check their biases. They're going to design their study to reduce biases. Okay, so that means staying open, trying to be objective in terms of what you might find. It might be contrary to what you expect. And you want to standardize everything as much as possible, so you're not communicating some subtle hints or information to the participant, right? To try to confirm what you already think. You want to be really as objective as possible. Placebo effect is something to be concerned about because I think we mentioned it's a real phenomenon. It causes physiological effects in our body. So if we think we're getting a treatment, we may actually somehow protect ourselves physiologically from getting sick, right? From experiencing something negative. Okay, so we want to always measure, always have a comparison or control group, and be aware of the placebo effect. Okay, a lot of these threats that I mentioned, some that I didn't mention, they can easily be counteracted by our design. So if we have a double blind, or we call a mass design, we can eliminate these threats. What does it mean to have a double blind study? If someone is familiar with that term, maybe like in a medical field?
If both the people who are doing the experiment and the participants of the experiment don't know who are taking placebo.
Yeah, perfect, excellent. So people who are doing the research or the study or the experiment, the researchers don't know who's in the treatment condition or who's in the placebo or who's in the control. And similarly, the people in the study, the participants, also don't know which one they're in. They're told oh, there are multiple groups that you might be assigned. You might get the placebo, you might get the active treatment, but you don't know. So your best guess is maybe 50-50, right? Yeah. So when that happens, it really eliminates bias, right? Or those researcher expectations from influencing the participants. Or the participants themselves trying to influence themselves, right? So it just really limits a lot of things. All right, lots of terms. I think, yes, I want you to know the terms, but more importantly, how important it is to consider all of this when you're designing an experiment. It takes a long time to plan it. You should be collaborating with others. You have an institutional review board, someone who's approving the study, who's gone over it, who's looked for all of these things. So by the time you actually conduct your experiment, right, you should have eliminated as many of these as possible. Okay. One more big threat to an experiment is measurement bias. So remember, part of empiricism, or how we conduct science, is through empirical methods with observation and measurement. Okay. So things can go wrong if we don't ask things in the right way. Maybe we're using terminology that's a little bit controversial, elicits certain emotions in people, right? Or maybe the actual tool we're using as a measurement is not valid or reliable. So that's what measurement bias is referring to, when the measure we're using is not accurate. Okay. There's many different kinds of validity and reliability. You don't need to know all the kinds now, just need to know generally what they are. If you go on in psychology, we'll talk way more about this. But measurements can be different combinations of valid and reliable. So there's these not valid, reliable, not reliable, not valid, valid, not reliable. Okay, so different combinations there. Reliable is the consistency of the measure. So every time you give someone the measurement, maybe it's a survey, they're responding the same way. Probably want some consistency, right? In measuring something like their personality, right? You're measuring the same thing, or what you think is the same thing every time. It's not drastically different. Validity is whether or not it's measuring what it's intending to. If we're interested in personality, it actually is measuring personality and not something else. What's something else that you think is similar to personality, but not personality, you might be measuring? Oh dear. Say it a little bit louder. Are you saying attachment? What? Go ahead. Okay. Ooh, okay. So you said some attitudes. I like that. It reminds me of like emotions or mood too, which we know fluctuate. It can fluctuate quite a bit even within a day. Yeah. So we're not just measuring if you're having a bad day, but we're actually measuring something like personality that's supposed to be enduring over time. It's apparent in many different situations that you put yourself in, many different contexts. Good. Okay. All right, so ideally, we want something that's highly reliable, highly consistent, and highly valid, has validity, which would be the upper right-hand corner. So it's the bullseye. It's consistent because all those points, those data points are together, so around each other. And it's in the middle. That's what we actually want to measure, right? The bullseye. Okay. All right. So this connects to operational definition as well. We want to make sure we have a clear operational definition so we know it's valid, right? It's measuring what we expect it to measure. Okay. All right, make sure you are reading your textbook because it's reinforcing all of this stuff. Sometimes we just spend a little time on it, but there's a lot more. Okay. This has come up before, so we're spending a little bit more time on it. It's the difference between internal validity and external validity. Okay, we just said, what is validity? It's how precise or accurate, right, our measurement is, or in this case, our study. Okay. There are different research designs that correspond to different levels of internal and external validity. Okay. So the highest internal validity, true experiments, that means that the researcher is in control, right? They can eliminate alternative explanations. They're very sure that the variable they manipulated is the cause to the effect. Okay? So because it's so controlled and standardized, it has high internal validity. Now on the other side, right, here we go from true experiments to correlational studies to descriptive studies. Descriptive studies have the highest external validity. So that means they're not artificial. They're similar to what you would experience in the real world. they will therefore generalize to the real world. We're pretty sure whatever we see in the descriptive study is going to be true in the real world. We're not so sure about that for a true experiment, especially if it's in a laboratory, it's very artificial. You're asked about hypothetical questions, but you don't even know what you do in the real world if you were put in that situation, right? Okay, so there's often a trade-off. Usually when we have high external validity, we have low internal validity and vice versa. So what we're doing is we're just noticing some of the limitations of research designs. No research design is perfect, but we're going to try our best to get the most accurate conclusions. And if we get it wrong, that's okay. That's what science is, right? We have more people doing research. It's self-correcting over time. It's iterative. It's a cycle, right? All right. How many people, I know it's not required for this class, but has anyone taken a statistics class before? Okay, good. I know not everyone. We did go over this a little bit in seminar. We'll talk about it for Monday folks today. I'm going to go over it fairly quickly, but let me know if you want me to slow down. Okay, we're going to now move from experiments to talking about correlational design. Remember, correlation is looking at the relationship or the association between two or more variables. The simplest one is with just two. And probably the most important thing to take away from this class is, correlation is different, not equal to causation. Remember, experiments, we can tell cause and effect. Correlation, we can't make that conclusion. All we can say is that they are associated, but not one is the cause and one is the effect. Okay. Lots of famous examples about something called spurious correlations, where it looks like two variables are correlated, but it's actually explained by a third or confounding variable. This is probably the most famous example, right? It's looking at ice cream sales over the year, right? Here's January to December. And then it's correlating that with violent crime. So how often, frequently violent crime occurs. Okay, this has been done with a number of different variables. You just put the variables together on a graph, and when they match, people get really excited. They're like, oh, that must mean that one causes the other, right? In this case, it doesn't make sense that one would cause the other. There's a third variable that explains this relationship. So what do you think it is? Crime and ice cream sales. Why are they both high and low at the same times? Yeah.
Both increase in summer.
They both increase in summer? Yeah, so it's saying right here in June and July, probably July really, it's like July, August is the highest for both. But why?
Because more people eat ice cream this summer. I don't know about July,
But more people are what?
eating ice cream this summer.
They are. More people are committing crime in the summer, and I see a couple of hands. I'm gonna ask you first. More people are active and out and doing things than when it's like freezing cold, especially if you live somewhere where it's like snowing or something like that. Yeah, that is the underlying similarity there. So it's not that these variables really have anything to do with each other than the time of the year and what people are doing during that time of the year. Okay. All right.
Not ice cream has something in it.
So you're talking about conspiracy theories now, which we're gonna be talking about in 240. Actually, I do have a really good example. This is probably the most harmful in psychology, in my opinion. Spurious correlation that's out there. And it was originally published in a very famous medical journal, was retracted because of bad science, but people have put things like autism correlated with, in the MMR vaccine, with literally like anything else that looks like it's increasing over time, and it looks like they're related to one, it looks like they're associated with one another. But usually what the most common similarity is just a lot of things increase over time just because of time. So I've seen people put like autism rates together with the increase of people eating organic food labeled organic, because we know people have been eating what we consider organic for a long time, and it looks like they're related, and so from that you could conclude eating organic food in bad science causes autism, right? Which does not make any sense. So we see this a lot. It's important to recognize correlation. It's not causation. Is there something else that might be influencing the relationship or explaining the relationship? Yeah. Sorry, I don't have the best hearing, so if I ask you to speak up or like repeat yourself, please don't take it personally.
Maybe the differences within individuals' vaccines compared to like the rates and the autism diagnosis, more likely corresponds to like medical treatment, and like people taking the hits to see like different types of doctors more often, increasing like popularity of technology for that, because the jump seems to happen like 70s, 80s, which would make sense.
Right, with the rise of like information technology, different kinds of technology. That's one of the biggest things that psychologists talk about, is just over time, we have more information. And so when you go to the doctor, they are able to maybe notice something earlier or get a formal diagnosis, whereas before they didn't have that opportunity. They might be labeled something else. Maybe there was an actual like criteria or diagnosis, or they didn't get any treatment at all. So a lot has changed in a very short amount of time, causing all of these simultaneous increases. Yeah, good point, thank you. All right. So just to say, I haven't already said it, because I literally talk about it in every class. I'm a developmental psychologist. They've done a lot of research, hundreds of thousands of kids over years. There's no evidence that there's even a correlation between the MMR vaccine and autism. The best, kind of the biggest explanatory factor is hereditary. So genes, they find that if you have a family member who has autism, you are more likely yourself to have autism. Okay. All right. So how are correlations measured? Just hopefully this is a review, but if not, correlations measure a couple things. One is strength and the other is direction. I think this was number 3 on your worksheet that you have. All right, so this is a nice visual because it shows both the strength and direction in one image here. All right, if we have a 0 correlation, that means there is no relationship between the two variables. If we move out from 0 toward 1, we're talking about a positive correlation. As one variable goes up, the other variable also goes up. The stronger we get, the closer we get to 1, the stronger that relationship, and vice versa for the negative correlation. Okay. A negative correlation means one variable goes up while the other one goes down. Okay. All right. So if you are ever reading a research study and you see that lowercase r, usually it's in italics and it's a statistic, and you see a number there, it'll range from -1 to +1. That is the correlation coefficient. Okay. All right. Often with the R, it's not up here, but you also see a p-value. Has anyone seen a p-value reading a research article next to R? Yeah, do you know what it's for, what it does?
Population size?
Close. Probability value. So it's talking about how statistically significant that correlation is. So it's a separate one, but you'll see them together often. You're looking for a p of less than 0.05 for statistical significance. Okay. All right. And the last thing in this little unit to talk about is research ethics. Okay. Remember in our first lecture we talked about psychology causing harm to people? Well, yeah, in many different ways, psychology and a lot of other fields have had a long history of unethical research. They haven't always got something called informed consent or let people know what's going to happen to them. Sometimes they were lied to. Sometimes just information was withheld from them. Experimenting on vulnerable populations, right, that had no power nor no control. So that's happened for a very long time. And it wasn't until fairly recently that the United States and places over the world have come together to create a code that, as psychologists, as scientists, we promise to adhere to. It's kind of like what the medical field has with their oaths that they take not to harm people. We have that for research, for practice, which is like the therapist relationship, right? And we have it for teaching too. Okay, so the one we're going to talk about specific to the United States is the Belmont Report. It came out in the late 70s. So it came out after the Stanford prison experiment, if you're familiar with that one. It came out after Milgram's obedience study where they pretended to shock participants until death, right? So all these unethical studies that are really famous and we talk about today, this code didn't come out until after all of that. So before that, they were literally just thinking, here, let me design this experiment. I think it's a good idea. People who think like me think it's a good idea, like let's do it, right? And then they would get into trouble where they would see, oops, I'm actually like hurting someone or harming someone. Like, what do we do? There's no oversight, there's no approval. None of that existed in any real way before the late 1970s. Okay. All right. So how many people have heard about Zimbardo or Milgram's study? Like maybe watched a documentary on it? Yeah, okay. It might come up some other time when we get to like social psychology especially. But I do want to talk about one particular study just because it really violates every single principle of the Belmont Report, which I'll introduce in the next slide. So this was not specific to psychology, but it's not unlike things that have happened in psychology. So it's the Tuskegee syphilis study. Has anyone heard of this study before? We use it a lot as an example because of just how atrocious it was. So this, it's not like it happened yesterday. It has been some time. It was before the invention of like penicillin to treat syphilis. If you aren't familiar with syphilis, it is an STI, sexually transmitted infection that we have an effective treatment for now, but it's actually kind of increasing again in the last couple of years. Even in like Humboldt County, like specifically here, so it's still around. What this study unfortunately wanted to do was to study what would happen to people who were not treated with syphilis. Like what was the natural progression of the illness? And for many people, it does lead to death, right? There's a latency period of like decades where maybe nothing's happening, and then you see these really severe symptoms of like organ failure, the way it impacts your brain to be like dementia, right? So really, really serious. You can pass it on to your children through like congenital ways, your partner, obviously, it's sexually transmitted. Okay, so one of the biggest problems with this study that they wanted to do was the population that they conducted the study with. So they chose, almost 400 people from the South, so they were African American. Most of them didn't have like high education. Some of them couldn't read, right? So they went to this population and said, do you want to be in this study, right? Here's all the great things we're gonna give you for being a part of this study. It sounded really good in terms of the incentives, right? They were getting free medical treatment, you're there getting some meals, transportation, they would get treatment for like minor illnesses, so like going to the doctor, which might have been a barrier for a lot of people. Even their survivors would get burial benefits. Okay, And they were not told basically any negative thing that could occur from participating in the study. Meanwhile, they were directly causing harm by doing spinal taps on them, like various types of treatments. They were not ever told they had syphilis. They were told they had bad blood. They were prohibited from entering into military where they could have got some additional benefits for their family because they had syphilis. And at some point during the study, penicillin came out as an effective treatment, and they were not given the treatment, even though it existed. So many problems with this study. There were some whistleblowers, but unfortunately, it went on for way too long. Like even until, oh, go ahead, you wanna add something?
I was going to ask,
Yeah.
did they have syphilis before they entered the study? Or
So yeah, that's good. So they were not like given syphilis, but they wanted, some of them might have had it, or some of them might have just contracted it over that time period, because it was something that was in the population. But yeah, that would have been just another thing if they intentionally gave them syphilis. Yeah. So it wasn't until like fairly recently, like the U.S. government has acknowledged that they did this, that they knowingly caused harm to people, and it was President Clinton a while back who like formally apologized, and they were given survivors were given benefits. But again, it went on way too long. It should have never happened. It is one of the studies that prompted the Belmont report that we have today. And just as I go through the different principles, you can kind of see where each one was violated in terms of that study. Okay, so 3 principles of the Belmont report. Number 1, at the top, we have to have respect for persons. The participants in the study have to voluntarily agree to be in the study and give informed consent. They have to know what they're getting into. They have to be told the basics of the study, right? The harms, the pros and the cons. They should have been told that they had syphilis if they did have syphilis, right? So this number was actually, I think altogether they started with 600 and at the end like 400 had syphilis, something like that. They started with a little bit more. Oops, wrong way. Okay. So respect for persons also has to do with vulnerable populations, like people who are incarcerated, right? There's special protections. People who are minors or under 18, there's special protections because they may not be able to give their informed consent in the same way. People with cognitive impairments, right? We need to treat these individuals with extra care because they are vulnerable. So when you submit a study and you're looking at a vulnerable population, there are extra precautions for what you have to do to make sure you can conduct your study. The second principle on the right, it's a little hard to, the color is very different on my screen. Okay, beneficence. That is, in general, we don't harm our participants, including animals. Now, does that mean like we don't cause them any stress or we don't harm them at all? No. We might cause them minimal harm, something that's similar to what they would experience in everyday life. That's generally considered okay. Or if the benefits really outweigh the costs. Maybe there's some animal research where we can really learn a lot, develop like this amazing treatment for cancer or something like that. We would consider the benefits of that for all of society, right? Okay. The last principle is justice. That's looking at who the participants are in the study. So it should be representative of the population. It should never be, we're gonna choose people who are in some way, right, vulnerable or don't have as much resources or for other reasons like have been historically taken advantage of or don't have power, right? It should never be like we're targeting those individuals for our study. You have to look at who it's gonna benefit and it should be representative of everyone. Okay. All right. So every study in psychology has to go through an approval process according to these 3 principles to make sure the study is ethical. In case you didn't know, in that informed consent, because you will be doing participating in some research studies as part of this class. In case you didn't know, when you fill out the informed consent, you have to be made aware of the purpose of the study. So what is it about in general? They don't have to tell you the hypothesis, they don't have to tell you very specific things, but you need to know enough so that you can decide, you have enough knowledge to decide whether you want to participate. Okay. You have to know whether you can be identified by them. Are they collecting any sensitive information? Could that potentially be leaked or, given to the wrong people? People you don't want to have that information. Okay, you should know that. You should know if there are any risks of participating in the study. Okay. You should know that at any time you can stop participating with no penalty, right? And just be like, I don't want to do this anymore, and walk out. That should be clear to you before you start. And if you want to skip a question, you're like, I don't want to answer that, I don't want you to know that, you can skip it as well. So these are participant rights that haven't historically been clear to people participating in research. And if you were, if some information was withheld for any reason, there needs to be a debriefing. So after the study is completed, they would tell you like, oh, actually the study was about something slightly different. We couldn't tell you at the time. Maybe we did a little white lie, but here's what the study's really about. That's called debriefing. All right, has anyone already participated in a research study here? Cool. All right, so make sure that you're gonna do at least 1 hour this semester. We use SONA, S-O-N-A. That's where people in the psychology department post their studies, and then you can sign up to participate in one. You will see an informed consent. It'll probably be really, really long, and at the end you have to say, I agree to participate in this study. Okay, yes?
Should you read the warnings or the whole thing?
It's not that long. It's not in terms and conditions. So it's maybe about like 2 pages or so. Yeah. I do, because I'm kind of interested in that, but yeah. I think it relates to this class, so I would suggest it. Okay. All right, any questions about psychological research? So as we go forward, we're going to talk about some actual studies. And now that you have some of the basic vocabulary, you'll be able to understand when we say, yeah, this was an experiment, right? Or this was a vulnerable population. They're doing minor, and we're looking at minors, right? You'll start picking up on some of that. All right. This time, let's see, Monday, Wednesday, we're talking about biopsychology. So we're going to start off. Now, we're not going to get very specific or detailed. If you are interested in things like genetics, there's whole classes outside the psychology department you could take. So this is kind of a very basic level, just letting you know that we won't go very deep into it. Okay, so we're starting off talking about genes. Here's a picture of Charles Darwin. Wasn't a psychologist, but has certainly influenced psychology, especially evolutionary psychology. And I just want to point out something, which is the idea of survival of the fittest. So that term gets thrown out there, right? Everyone probably has heard of that outside of your studies. But there's often a misconception about what that means. So some people think that means like only the strongest people will survive, right? What does it actually mean? Someone who's familiar? Yeah. Yes, the ultimate goal for us as humans, as a species, is for us to survive, and make it more specific, like if we have offspring or even our kin, right, we want them to survive long enough so they can pass on their genes, right, so that our species continues. And the most important thing of what you said is you need to be able to adapt to your environment. could be any different kind of environment, right? But those who are adapting to their environment are the ones who are surviving and passing down their genes. Okay, so think about that in terms of psychology. It could apply not just to like features, like how big we are, how big our brains are, but it could refer to like behaviors as well, right? Whether someone is more bold or someone is more timid, which can relate back to personality in some ways. Okay, so in certain environments, some individuals will survive, others won't. What's most important is to adapt to our environment. Okay. Good, so keeping that in mind, thinking a little bit back to just what we're given in terms of our genes when we're born. Right, so we get 23 chromosomes from our bio mom, we get 23 chromosomes from our bio dad. That comes together to be 23 pairs of chromosomes. What does that mean? Well, each chromosome here, right, is within a nucleus. We know the nucleus is within our cells, very important part of our cells, right? The chromosome is this kind of X-like sausage structure is how they describe it. And it has these very long strings of DNA. So genes are very specific locations along those really long strands of DNA. So we can pinpoint certain places in our DNA that correspond to different genes. Okay, and each gene has basically instructions or directions for making proteins, right? Corresponding to different areas of our body, different functions that we have, so that we can both grow and sustain life. So it's telling our body what to do within that genetic material. Okay, and when we say, we're talking about genes, if we hear the word allele, that means a variation of the gene, and that's just according to what we're given at birth, right? What genetic information is passed down to us from our biological parents. So we may have one version of a gene rather than another version of a gene, just what we're born with. Okay. How many people, like, I remember this. This is like review for me. Okay, good. If not, lots of study material on Canvas in your textbook. Okay, so some more terms. We have genotype. Genotype refers to your genetic inheritance, again, what you were given genetically by your bio parents. It's the potential, so it's what could be. Whether or not it actually comes to fruition, that's something different. That's phenotype. Okay. So phenotype is what we actually can like observe about a person. So maybe something about their appearance, their personality, their intelligence. It's what actually happens in real life. And it's more than just your genes, because now it's considering environmental factors, family, culture, your resources, right? So they don't always equal each other, they are different, but definitely phenotype consists of the genotype, but more. All right, we'll get to some examples in just a bit, how they combine, yeah. Let's look at these different ways that genes combine. So again, we have lots and lots of genes, lots of genetic material. And they combine in different ways. So one way they combine is called additives. Basically, all of the genes have an individual contribution. It all adds up to produce the phenotype. Okay. Something like height is composed of hundreds of genes. So all of them contribute, beep, 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 beep. Comes out at the end of that is how tall you are, how tall you can be, the potential, right? Hair curliness, same thing. Skin color, right? All of these genes are individually making contributions for the overall product. Probably the combination you're more familiar with is the dominant recessive. Nothing personal. Yeah. So dominant recessive hereditary is looking at when you have not an additive effect, it's not an additive, but instead, usually there's one that's more influential than the other. So the dominant is usually more influential than the recessive. And many times the dominant gene completely overshadows the recessive gene. There's like no indication that it's even there, exists. But in other times, the recessive gene might still have some impact. It might come through a little bit. We call that incomplete dominance. So here's an example of like, yeah, you might have a recessive gene for like the lighter color in the chicken feathers, and it might show through a little bit, right? Where you don't have a completely black or completely white chicken, but you have kind of a mix of both. We see this with eye color as well. If anyone's familiar with like hazel eyes, that's where we see some incomplete dominance coming through. All right, so we're talking about dominant recessive genes, combining different alleles or variations. It's very, very rare that any trait or thing is due to one gene, like monogenetic. That rarely, rarely happens. Almost everything is polygenetic, meaning again, the genes are coming together in either that additive or non-additive way. And just some different terminology for you. We're given genes from different bioparents, and when they are similar, the same, we call that homozygous, and when they're different, we call that heterozygous. So that, just different combinations that can happen. Okay. Probably the classic example is eye color, right? Although it's much more complicated than what is presented here. Again, because of incomplete dominance, as well as environmental factors can also influence our eye color. Any guesses how? What do you think?
I don't know if this is just a myth, but like the amount of sun exposure, like
It's a good one.
that you have, I guess.
Yep, and your hair too.
Yeah.
Yep, perfect. Yep. So there's a lot of influences. Okay. And let's see, we'll do one more slide today. So finishing up kind of interactions. So far we've been talking about gene and gene interactions. Let's now introduce more about the environment. So gene-environment interactions. Think back to those terms, genotype and phenotype. So one of the reasons we see phenotype is because of the range of reaction or reaction range. What that means is we are all born with a genetic potential, a range, but where we end up will depend on the environment. Okay. So let's take that example of height. Okay. So let's say our genetic potential for height, how tall we could be just based on our genes, is 5'8". Okay. Will everyone get to be 5'8" who they have that as their genetic potential?
No.
No, yeah, they might end up here or here instead, right? Why? What are some reasons why they might not meet that height potential?
Maybe they drink a lot of caffeine.
Okay, so maybe things that they're consuming could be like something that's toxic or like would stunt your growth, maybe caffeine in particular.
What they're not consuming.
Say it a little louder.
What they're not consuming, like if they're not getting enough.
Oh, if they're malnourished? Yeah, so if they're not getting all the vitamins and nutrients that they need, yeah, that's a huge one historically, right, over time. Anything else? Yeah, perfect. So something like a medication. Have you ever heard of like, oh, go ahead.
Problems with pregnancy.
Yes, so thinking about in utero, what's that environment like? Yep, that's a really great one. I also thought about like people who unfortunately push their bodies too far in terms of like athletes and things like very early on. That could lead to additional, combined with malnourishment, like other kind of stunting that we would see. Yeah, good, okay. And then this last term here, epigenetics. If you aren't already familiar with this term, what it means is you have your genes that you're born with, your genotype, And then you might experience something in the environment that might turn on or off certain aspects of your genes. So think about a light switch, like turning something on, turning something off based on your environmental experiences. A really good example is schizophrenia. So we know there's a genetic susceptibility to schizophrenia, right? If your parents had it, you're more likely to have it. However, there's also thought to be usually some important triggers that have to happen, usually in late adolescence, early young adulthood, like stress, like the use of substances, like marijuana, whatever it is. If you have that particular combination, you're much more likely to develop schizophrenia, maybe than someone who didn't have those environmental triggers. Okay, so we call that epigenetics. It's very important concept. All right, we are out of time, so I'll see you on Wednesday. We'll start off with the nervous system.